the University of Toronto page, which reads as follows. After a brief stint as a filmmaker and an aspiring successor to Ingmar Bergman, Francois Truffaut, and Michelangelo Antonioni, Dr. Marshall obtained his medical degree from the University of Toronto in 1977 and changed the medium of his editing passion from celluloid to flesh. He completed a fellowship in general surgery at Dalhousie University, Halifax in 1984, and undertook a research fellowship at McGill University under the mentorship of Dr. Jonathan Meekins, following 3 years of a critical care surgery practice in Halifax, culminating in the receipt of the Royal College Medal for Surgery in 1989. He moved to Toronto in 1990. He spent 15 years at the Toronto General Hospital as a critical care surgeon and intensivist before moving to St. Michael's Hospital in 2005, where, in addition to the above, he reawakened a long-standing interest in trauma. Dr. Marshall is currently a professor of surgery at the University of Toronto. He runs a CIHR-funded laboratory at St. Michael's Hospital focused on the mechanisms of prolonged neutrophil survival in sepsis. In addition, he serves as chair of the Canadian Clinical Care Trials Group, the oldest and most productive investigator-led critical care clinical trials group in the world. He is a past president of the Surgical Infection Society and past chair of the International Sepsis Forum. And Dr. Marshall has published more than 210 papers and 75 chapters and given more than 600 invited lectures around the world. So, of course, it's a great privilege to have him here speaking to us today. I'll just ask everyone just to please mute yourself during the presentation, and there should be time at the end for questions. And with that, John, the floor is yours.
Okay, well, thank you so much for that introduction. It is a little bit out of date. I was the chair of the Canadian Critical Care Trials Group until 2012, so the current chair is actually Kusum Menon. Still kind of one of the main things in my life, one of the real joys in my life. But I really appreciate the opportunity to share some thoughts with you today. First of all, I want to, if I can do this, declare any conflicts of interest, and there really aren't any relevant to what I'm going to talk about, except for the fact that I'm the Canadian principal investigator in the REMAP-CAP trial. And as will become apparent, I think, during this talk, I'm a real enthusiast about the platform trial model and what it might mean for, particularly for investigator-led research in the future. And it's that I want to really kind of explore today. So the history of clinical trials goes back many centuries. In fact, there are allusions to things that looked like trials 1,000 years ago. But probably from a Western perspective, the first real attempt to understand biology by randomization was this experiment that was conducted by James Lind, who's was a ship surgeon in the 18th century, time that scurvy was a major problem for sailors. And he got the bright idea of seeing if there might be a cure for surgery. He tried 6 different interventions that included cider, sulfuric acid, vinegar, seawater, citrus fruits, and barley water. And so what he did is he assigned 2 sailors to receive each of these for a period of time. And over the period of time that he was doing that, some of the sailors got worse, some of them improved strikingly and took to helping some of the other sick sailors out. And the ones who improved were the ones who had citrus fruits, obviously. And although it wasn't recognized at the time, what he had shown was that something in the citrus fruit, specifically vitamin C, could prevent scurvy, or conversely, scurvy is a vitamin C deficiency. But the important thing here, I think, is the concept that we can actually test the real world, and the most powerful tool to do that testing is randomization. The contemporary model of the clinical trial, the one that I think most of us are most familiar with, dates back to the 1940s and the work of Bradford Hill, who was studying streptomycin for the treatment of tuberculosis. And he conducted what in many ways was the first contemporary clinical trial and introduced some key concepts in trial design. Again, randomization was critical, a randomization because it could control both known and unknown confounders, and therefore produce the greatest probability that an effect was attributable to the specific intervention under being studied. He also introduced the concept of blinding the interventions to avoid bias, and concealment of allocation to prevent the trialists from kind of knowing what the next intervention might be, and therefore selecting patients based on the prediction of a response or non-response. So that has really been the model that we have used over the ensuing 70 or 80 years, is a conventional model of a clinical trial that is focused on a specific intervention. In Brevard Hill's case, it was streptomycin for tuberculosis, but it is many other things right now. The key factor here being that usually clinical trials study one intervention. They study it in a population of patients that have that disease, and in designing the trial, they start with a prediction of what the size of an effect might be and what the direction of that effect might be. And as a result of that, they're able to come up with an estimate of the size of the population they have to study to achieve that effect, if the effect is true. And that kind of informs a key element of the trial design. And the ultimate result is a dichotomous conclusion. I think we've become addicted to the concept that trials answer a question with a yes or no answer, and we put that the kind of cutoff mark for that at a probability of less than 0.05. So here's an example of a conventional clinical trial, a really well done clinical trial. It was done by the Canadian Critical Care Trials Group, published in JAMA a couple of years ago. And the question being asked here was, can probiotics, specifically Lactobacillus rhamnosus GG, reduce the rates of ventilator-associated pneumonia in intubated critically ill patients receiving mechanical ventilation? So single intervention, a population of patients at risk for ventilator-associated pneumonia by virtue of being on mechanical ventilation. So to do that trial, you have to identify that population. And so we came up with inclusion and exclusion criteria and estimate the effect size. And based on a review of the literature, we assumed the VAP rate would be 15%. And what our hope was to reduce that by a 25% relative risk. And so doing a simple sample size calculation said that we need to get 2,650 patients to do that if we want to have 80% power and an alpha level of p less than 0.05. This is the kind of thought process we all go through in planning a clinical trial. And it is the basic architecture of a frequentist trial design. But conventional trials are, I think, as anybody who's done them will recognize, they have a lot of shortcomings. They're very time consuming. The PROTECT trial or prospect trial of probiotics was more than a decade, or about a decade, in evolution, and probably about 4 years in patient recruitment. They're expensive, and that expense involves not only the recruitment of patients, but setting up the trial to start and closing down the trial at the end of that time. They're also inefficient because they only give you one answer to one question, and they're critically dependent on your having asked that question correctly. And frequently, the answer that you get is indeterminate. It's anytime a trial is evaluated, whether you do or do not meet the p-value you set, there are always reasons to think that the answer might be otherwise. And so you're left with a lot of money and a lot of effort being spent on something where the answer is still not necessarily definitive from a clinical perspective. So there has been a lot of thought about how can we be more efficient in running clinical trials. Obviously, one strategy would be to do a trial where you study two different interventions at the same time. And that's the essence of a factorial design, where you have treatment A and treatment B, and a patient who's randomized into the trial can be randomized to treatment A or treatment B, or both, or neither. And these can then be evaluated independently. And so in a sense, you get two trials for the price of one. Another option would be to run a clinical trial and, rather than shutting it down at the end, use that same framework for studying the second question in sequence. And another option that has become quite popular, I think, in the investigator-led research community is to promote co-enrollment of patients into clinical trials. We began to think about this process in the Canadian Critical Care Trials Group a couple of decades ago, primarily because we were faced with an increasing number of trials and the potential for conflict between trials and competition for patients. And so the question obviously arose, do we really have to follow a dictum that says we only enroll patients into one trial at a time? And we studied the issue of co-enrollment. And as we looked at it more closely, it became apparent that co-enrollment was not only more efficient, there were all kinds of reasons why it was probably preferable to encourage this kind of attitude of research in ICUs that were participating in research, and that families were quite comfortable with the concept that their patient might be co-enrolled into several trials. So that's another approach to trying to make trials more efficient. Now, more recently, and this is probably in the last decade or so, a few other models have emerged that add some details to how to make trials more efficient, and also perhaps how to focus them more specifically on the populations you're most likely to benefit. This was an excellent review of that published in the New England Journal of Medicine a few years back. And what I'm going to focus on primarily here is going to be the platform trial. But I want to talk about two separate aspects of the evolving trials landscape. One is the design of trials, and the other is the analytic framework that is used within those particular designs. So one model of a master protocol is an umbrella trial, which is in a sense a variant on a factorial design. You take a single disease and you randomize patients to more than two different possible interventions. So in this particular schema from that article, you're looking at a single disease and multiple potential targets in that disease. If you identify one biomarker positive, then the patient gets targeted therapy 1. If biomarker 2 is positive, they get targeted therapy 2. If 3 is positive, they get targeted therapy 3. And if they have 2 of them, for example, 1 and 3, they could be randomized to both interventions. But what that actually allows you to do is to study several different interventions based on the biomarker delineation of a potential to respond to treatment. So the umbrella trial is a model that studies multiple interventions for a single disease. It's become very popular in oncology, breast cancer, for example, where there are multiple different biomarkers that predict response to therapy. It's a very attractive research model. In a sense, the corollary to the umbrella trial is the basket trial, where you look at a particular intervention which may have value across multiple different diseases or subgroups of a disease. And so you're screening for the presence of the target in a variety of different disease populations. And if they have that target, they're enrolled into the trial. So your focus here is on the intervention again, but you're not being bound by a single description of a disease type. This also has found a role in oncology, and particularly for checkpoint inhibitor studies where abnormalities in these are found across a number of different histologic types of cancer. And so these are being combined together into single clinical trials. The platform trial is, in a sense, an expansion of the umbrella trial. So a platform trial, and it's a bit of a misnomer because although it is a trial, it is more fundamentally a platform that multiple trials can be conducted on. So what you do is you identify a broad population of patients. They could be patients with a common disease, a common risk factor. So the disease could be ventilator-associated pneumonia. It could be the risk for ventilator-associated pneumonia. But you want to have a fairly broad characterization of this population. Then you randomize those patients to as many different interventions as you want. It could be standard of care. It could be 2 different interventions as shown here. You run the trial, and as the results become apparent, you adapt the trial. You finish one arm as the trial reaches its endpoint, but you can also add new arms as the trial continues. And so what you've now got is a clinical trial that can study multiple things at the same time, and in fact can serve as a trial in perpetuity as you add new arms as the trial goes forward. This, in many ways, is the model that was used by the recovery trial in the United Kingdom. I'm going to talk more about the REMAP-CAP trial, which added a few other nuances to the platform trial model. But at least in recovery, they had a priori estimates for effect size that led to arms being dropped, and had the potential of bringing in new arms as the biology of COVID-19 was understood and as trial conclusions were reached. So the way, at least in REMAP-CAP, that we have kind of framed the concept of a platform trial is that you start with the condition of interest. And one of the things that I think is becoming very, very clear is that this is going to be critical in the future to really understand what that condition is. We in REMAP-CAP set it up to study interventions for future pandemics, and so focused on ventilator-associated pneumonia. There's a great platform trial called Practical, which is looking at hypoxemic respiratory failure. But I think what you want to have is something that is a clinical phenotype that's broad enough that you have a lot of patients. But one of the problems that's going to occur is these are going to overlap, and how do different platforms interact with each other when there is an overlap in the conditions. And I'll say a little bit about that towards the end of this talk. But having identified that condition, you then develop a master protocol, which essentially is the skeleton of the trial. It identifies the entry and exclusion criteria for the population you're going to study, outlines the design elements for the clinical trial, and specifies the particular outcome that you're going to look at. We then have different domains, and a domain is a therapeutic area where you have more than one option. If you look at community-acquired pneumonia, for example, we had an antibiotic domain, a corticosteroid domain, an immunomodulatory domain, a mechanical ventilation domain. Each of these are discrete areas in the management of a disease whose management is multifactorial, and where there are discrete treatment options within each of those. So you start with a certain number of domains, and you then, within each domain, you have interventions. And the interventions may be usual care. That's certainly what we've been doing in REMAP-CAP, or they may be an experimental agent. So in antibiotic domain in REMAP-CAP, we might have usual care, antibiotic A, and antibiotic B. There's an opportunity within this to even stratify patients further. For example, on the basis of a biomarker, whether the biomarker is positive or not positive. Again, in REMAP-CAP, in our corticosteroid arm, we stratified patients based on whether they were in shock at the time for receipt of corticosteroids. So what you have here is kind of a multi-layered protocol that allows you to hone in fairly specifically on discrete populations of patients at the same time as you're studying multiple other things on the platform. So the way it then proceeds is that you set in advance criteria for concluding the trial for success, futility, or harm. And as you reach those endpoints, that particular domain is ceased. You may bring on new interventions into that domain, perhaps 2 new antibiotics, once you've shown that one of them is inferior, for example, or you may bring on new domains. And they can be brought on really at any point during the clinical trial. But what you have here is kind of an ecosystem in which a particular disease is being studied with multiple different aspects of it being evaluated at the same time, and these changing over time as you learn from previous patients who are enrolled in the trial. What it looks like in terms of paperwork is you have a master protocol, you then have domain-specific appendices, and these will outline the specific entry and exclusion criteria for the particular interventions that are being considered in that, and they are often more they're not necessarily the same things as in the master protocol. But to be eligible for that particular domain, you look at what the domain-specific criteria are. And we also in advance develop a statistical analysis plan for that domain that outlines how the data are going to be evaluated once the domain has been concluded. Okay, so that's the kind of architecture, the trial design. I want to just look a little bit at the analytic framework because certainly there's the big difference between remap cap and recovery is in the analytic framework that we've been using. Here is the conventional model that has been used, which is a frequentist model. And in that model, in the prospect trial, there was no evidence of any benefit for lactobacillus. The p-value is greater than 0.05. In fact, it was 0.73, and the likelihood was, if anything, the point estimate was on the side of harm rather than benefit. But this is all very, very arbitrary. And we've become, I think, accustomed to confusing a p-value less than 0.05 with the truth. And of course, it's nothing like that. So what does a p-value of less than 0.05 actually mean? Well, it's the thinking for this goes back to the time of Ronald Fisher. And essentially what a p-value means is the likelihood that you see this result that you saw in this particular trial, or a result that's even greater, on the assumption that the treatment is ineffective. So you're actually starting from the assumption that there is no effect here, and you're trying to reject that assumption by showing that it's very unlikely that you'd see this value or a more extreme value on the basis of random chance. That's kind of difficult to get your mind around, and it's not entirely the kind of way that as a clinician you approach the management of a patient. We're much more interested in understanding what's the likelihood that this is going to benefit. And the Bayesian model, Bayesian analytic model, approaches this, I think, much more effectively. This is thinking going back to the 18th century as well, Thomas Bayes, where the underlying concept is you're looking at a distribution of probabilities and trying to understand where in that distribution a particular outcome lies. That distribution of probabilities is impacted not only by the observed data, but also by what you already know about the substance, already know about the subject in advance of undertaking the clinical trial. And that's a fundamental difference. We make the assumption in a clinical trial that we know in a frequentist trial that we don't know anything about it and that the result is a complete surprise and that we're absolutely neutral. You don't have to do that in Bayesian inference. You are coming up with an estimate that the intervention is likely or not likely effective. So, this is called the posterior probability at the end of the trial, and all that is the probability that the therapy is effective. This, to me, is what a clinician does. When you look at a patient, you're not interested in knowing with greater than 95% probability that what you're going to do is going to help them. I would be pretty happy if I knew that there was a better than 50-50 chance that a difficult decision was going to be beneficial. And so the Bayesian framework, I think, better reflects the clinical decision-making framework. So, frequentist trial recruits patients until the hypothesized effect size is likely. A Bayesian framework, you just recruit patients until an a priori specified outcome is achieved. So a lot of work is done in advance of the trial to kind of model the different potential outcomes and to figure out how you're going to determine when you've reached an endpoint. That's all done a priori, and then it's the data that are accumulating that drive the conduct of the trial. So adaptive trials change trial elements in response to the occurring trial data. You can change the sample size based on the event rate as you see that. You can change doses or subgroups. And you can change randomization proportions. And this, I think, is one of the really intriguing, although complicated aspects of an adaptive clinical trial. So the way this works is you've done a lot of pre-trial modeling. and you've created these algorithms and that provide rules for what you're going to do. If you see this, then you're going to do this. If you see this, then you're going to do that. And you let the trial run, and the trial just continues to run. And as the trial goes along, the data are generated, and you look to see if a stopping rule is met. If you've met a stopping rule, you stop the trial and begin the next phase and report the phase that's been stopped. If you haven't, you revise allocation and sampling rules based on the algorithm and continue data collection until you do, and that one of those things may be a conclusion of futility. So for the randomization process, this is called response adaptive randomization. And this schematic shows how that works. So essentially what you're doing is you're saying, as the trial goes forward, you're generating information about which things seem to be doing better and which things seem to be doing worse. And as a consequence, you're going to change the randomization proportions so that patients are more likely to be randomized to those interventions that seem to be doing better. So you start here, this is a hypothetical example, with usual care and 3 different drugs. You're constantly monitoring results, and you see at the first analysis, the first adaptive analysis, that standard care is doing slightly better, as is drug B. And so more patients are now being allocated to those particular groups. And conversely, fewer patients to drug A and to drug C. 700 patients, it's only these 2 options because the others have been shown to be less than these 2. And as you move forward, you find that at the end, at 1,100 patients, all patients are getting drug C or drug B because it has proven to be superior to the other 3 options. One of the things that's kind of neat about this is that as the trial goes forward as a perpetual platform, this can then become the standard of care for all subsequent patients recruited to the trial. So in a sense, you have a mechanism that is translating knowledge as it is accruing that knowledge. So it's kind of a hybrid quality improvement clinical research model. So as far as the conclusions go, domains are analyzed independently without reference to the other domains, although it is possible to actually use data from other domains to add confidence to the control group observations in the trial. And you stop the domain when a priori stopping boundaries when that. In conventional RCT, the results are reported in a manuscript for that particular intervention on that platform. So in REMAP-CAP, the way it works, our criteria are shown here, that for superiority, it's a posterior probability of 99% that the odds ratio is greater than 1.2. For inferiority, posterior probability of 99% that the odds ratio is less than 1. And for equivalence, a 0.9 probability that the odds ratio is between 0.8 and 1.2. And so the trial for each domain continues until these stopping boundaries have been met. So rather than a it works, doesn't work conclusion, we come up with the probability that the intervention was effective or was not effective. So just coming back then to applying this to how one might think about redesigning trials when one's doing them as a platform rather than a single trial. If you look at this fundamental question in the ICU, what's the best way to prevent ventilator-associated pneumonia? Probiotics was a good question, and that could be a domain on a platform. Other people have asked the question, what about positioning? What about selective digestive tract decontamination? What about early feeding? You could actually evaluate each of these on a platform and independently on that platform. As you reached a conclusion, then you could look at adding new domains to that particular platform. So this is attractive not only because of the efficiency with which you're able to conduct the trial, you're also able to understand interactions between domains and if with enough patients. And so you're again modeling the clinical reality of the treatment of a condition where you may use multiple things, early feeding, GI prophylaxis, and positioning, and you're able to see how those different variables interact with each other. So I think in many ways, and particularly for prophylactic strategies, this model of a platform trial really has a lot to argue for it as a more efficient means of conducting clinical trials. Another feature of the platform trial, which I find particularly attractive, is that it is quite straightforward for platforms to collaborate with each other. During the pandemic, we had three separate platform trials, ATTACK, REMAP, CAP, and ACTIVE 4 with the NIH that got together to study anticoagulation. And as a result, by combining the three platforms into a single trial, they were able to recruit over 3,000 patients in a matter of about 5 or 6 months and publish data on heparin, full dose heparinization in COVID-19. So the way that we came to the platform trial model with REMAP CAP actually goes back to the early pandemics of the 21st century, SARS and H1N1, where the clinical care community wanted to generate answers, but found themselves overwhelmed by the complexities of trying to actually launch trials during a pandemic. And so there were very few clinical trials, and certainly no adequately sized trials done during those crises. Following the H1N1 pandemic, we organized a colloquium to discuss this theme of pandemic preparedness. And this brought together groups from INFACT, which is an international group of critical care research groups, and ASARC, which is an international body of infectious disease public health research groups, to discuss what should we do to be better prepared for the next pandemic. And the big concept that came out of that was that if you were going to do a trial during a pandemic, the trial had to have been designed and up and running in advance of that pandemic need. And so that was where we got interested in the concept of the platform trial, started to develop REMAP CAP as a platform for the next pandemic. We had recruited probably slightly less than 100 patients by January 2020, but did have it funded in the EU, in Canada, and in Australia. And then in January 2020, much to our chagrin, we found ourselves faced with having to respond at a time where we were still trying to work out some of the glitches in the model. So here's where REMAP CAP stands now. It was designed to study patients with community-acquired pneumonia because that's what a pandemic infection looks like. And it was pragmatic in its design. Entry criteria really pretty in a non-pandemic model, but we had a kind of sleeper pandemic model ready to go when the need arose, and that was activated in late February or early March of 2020. To date, we've recruited over 13,000 patients into the trial. We have 18 different domains that we've either completed or are active now, 66 different interventions, and multiple publications. So we've had 4 in the New England Journal, 7 in JAMA, and 3 in other journals. We have sites really across the world. We've started a site in South Africa since this map was drawn, sites in Japan, and we're trying to expand into Latin America. Most of our activity is in Canada, the United Kingdom, Australia, New Zealand. But interestingly, we've got very strong engagement in Southeast Asia, and particularly in Nepal. So we are a global clinical trial. With the pandemic, we further refined the model so that we had 2 states, a severe state, which was the equivalent of being in an ICU. So this was for patients who received positive pressure respiratory support or vasoactive agents. If you didn't, but were hospitalized, you were said to be in the moderate state. And so patients were stratified between these 2 different states in the trial. We also changed our primary outcome from 90-day mortality to organ support-free days over 21, where mortality gave you a value of -1. And the reason for this modification was to have an earlier endpoint that could drive response adaptive randomization. And so this is how the results are, how we present the results as kind of a heat map where dark red is death, dark blue is no organ support, and you can kind of see the proportion of patients in each of those different boxes over the period of time that the trial is being run. And we can generate a odds ratio that informs our conclusions. So another thing about this, about the platform trial that proved really helpful during the pandemic is the fact that we could add new dimensions as we learned about the disease. So it became apparent quite early on that thrombosis was a major complication of COVID-19. And so we added a couple of domains, an anticoagulation domain, an antiplatelet domain. Other data had suggested that statins might be beneficial, so we added a statin domain. Vitamin C, there was some enthusiasm for vitamin C and ARDS, and so we added the vitamin C domain. We were able to stop our antiviral domains quite early because they didn't work. When it became apparent that the virus binds to ACE2, we added a domain looking at ACE2 or RAS inhibition. And these were added as the trial moved forward. And apart from the needing to build the database, that was not particularly complicated. So here were some of the findings. We found, interestingly, that a differential response for therapeutic anticoagulation with heparin, depending on whether you were moderately ill or severely ill. If you were moderately ill, as shown in the lower left-hand corner, you did better with anticoagulation, less red and more blue. If you were severely ill, it was actually the other way around. We showed, here's data we just published last fall on simvastatin. This was close to 3,000 different patients randomized to simvastatin. And overall, the odds of organ-free or organ support-free days, the probability was 95.9%. That was less than our endpoint. We stopped it because the pandemic was quieting down and we hadn't reached a firm endpoint. But certainly from a clinician's perspective, it suggests, I think, quite strongly that simvastatin may be beneficial. And so make of it what you will. Certainly it's a theme, I think, that merits further investigation. These are our results overall. We found quite early on that hydroxychloroquine was actually harmful in seriously ill patients, as was lopinavir ritonavir, and you put the two of them together and they were not, they were definitely harmful. Corticosteroids, we also saw benefit for corticosteroids. We used hydrocortisone. We showed improved outcomes with IL-6 receptor antagonists, either tocilizumab or sarilumab. No benefit for IL-1 receptor antagonist. This differential response for heparin, depending on the state. No benefit for convalescent plasma, although there was a suggestion that in immunocompromised patients, it might be beneficial. And we're currently testing that right now in a continuation of the convalescent plasma domain. No benefit for aspirin or P2Y12 inhibitors, although in our analysis of data at 6 months, there's actually a survival signal for the receipt of these signal for benefit. Probable harm with vitamin C, harm with ACE2 RAS inhibitors, and the probable benefit for simvastatin. So over, this would be 10,500 patients with COVID-19. We were able to draw quite a few conclusions that really have impacted the management of that disease. And what this is doing is kind of opening the door to thinking about how can we better marry research and quality improvement in a learning healthcare system. And I think, to me, the exciting thing about the platform trial is that it actually shows a path forward to do this. So in the United Kingdom, for example, hospitals are funded to do research and expected to do research. So the British government has adopted the perspective that research is a part of the provision of care and it needs to be funded out of healthcare dollars. That was in no small part the reason that recovery was so successful. And frankly, it was also the reason why REMAP-CAP was successful, because over half of our patients actually were recruited in the United Kingdom, where the infrastructure to do these trials was already in place. They, during the pandemic in the UK, prioritized trials, and they designated which trials would be studied, would be used, would be the preferred trials for COVID-19 therapies. You can see recovery for hospitalized patients and REMAP-CAP for ICU patients, but a whole kind of portfolio here of trials, depending on where in the disease your illness was at the time. We are working right now with the World Health Organization on a somewhat similar concept. One of the big challenges during the COVID-19 pandemic was the provision of oxygen. Even in well-resourced settings, ventilators were in short supply, and even oxygen supplies were sometimes challenged. In low and middle income countries, this is a huge, huge challenge where oxygen often is generated through oxygen concentrators and tanks just aren't available. So what we are proposing is a globally federated research platform that will study multiple different interventions to optimize oxygen delivery for patients with severe respiratory infection. So this could be anything from the target saturation to different modes of mask ventilation, to different kinds of mechanical ventilation, even to ECMO, or to pharmacologic management that might improve oxygenation without altering the way that that oxygen is actually delivered. And this is still in the very early stages, but we have expressions of interest from over 100 people in groups around the world, and a fair bit of support within the WHO at some of its higher levels for the idea of trying to build this globally federated trial where everybody who's doing studies of respiratory support for patients with respiratory infections would collaborate on a single research platform and share their data. So I think what really the platform trial offers an opportunity. COVID-19 has really accelerated that opportunity for us to move beyond evidence-based medicine to practice-based science. In other words, to move beyond thinking of the generation of evidence as being something outside of the provision of care to thinking about the generation of knowledge as being one of the core elements of the provision of care to patients. And so I think that to me is what's really exciting, the potential transformation of the healthcare system that this particular model potentially promises. So I'm going to stop at that point and be very happy to open the floor to any discussion that you might have.
John, that was
Thanks so much, John.
amazing.
So we will invite any trainees on the call to go first if anyone has any questions. I know Jeannie was keen to jump in there too, but I don't see any other hands. So Jeannie, you go ahead. What's your question?
I have some questions about the funding of these trials. So, you know, you go to Health Canada, say, okay, we're going to do a platform trial. We don't know how many patients we're going to need to enroll in this trial. Right. So we can't give you the normal budget. And I think that that's causing kind of havoc.
Yeah.
We had a recent one where we just wanted to do the Canadian portion for an Australian platform trial. Mm-hmm. So we were going to Health Canada just for a limited number of patients. So that's like, okay, we just want money for 300 patients. But then they said, oh, we don't like the statistical analysis plan by the Australians, but the Australians are already underway with their trial. Mm-hmm. Like you can't change the math halfway through the trial. So like, you know what I mean? Like just navigating the funding issues around these trials.
It's a huge, huge challenge, and it's completely unsolved at present. How we are working REMAP-CAP, we have multiple different funders. They're regional funders by and large, but we're also in discussions with CIHR because CIHR has, through their clinical trials program, has recognized that they have to figure out how to fund platform trials, and they don't know how to do it right now. I think that this is contingent on us as investigators to come up with models that they can then adopt. I don't think that the nuances, the importance of collegiality, there's a whole host of things that come up with this trial model. And I don't think that unless you're part of that system, you can do that. So we've done a couple of things. We had a colloquium, sort of a follow-up colloquium on the lessons of the COVID-19 pandemic. And one of the lessons that's come out of that has been the need to engage with funders and policymakers about supporting research networks as a model for delivering clinical trials and coming up with new funding models that would fund platforms. And then you could, you might apply for funding for specific domains on that platform, but that you would have the platforms. But that means that rather than us competing against each other to get our own platforms funded, we need to figure out how do you take the healthcare landscape and break it down into a bunch of thematic areas that can potentially overlap and work together, and that those thematic areas can be funded as platforms, and then individual domains, even in a competitive manner, could be funded as trials. So we're having a meeting actually in September, an international meeting, to kind of discuss how platforms can work together. And that's clearly one of the issues here, is how do funders start to think about this? It has to happen soon, because as platforms are being funded, it's kind of setting in stone a model. And if that model is not right, we're going to find ourselves with a very messy situation a few years down the road.
Yeah, I don't know if you can still raise your hand in a Zoom call, but fantastic presentation, and it is pretty compelling. And I guess I'm just thinking in terms of arguing with CIHR that it's worthwhile investment. It sort of begs the question: has anyone looked at a cost effectiveness assessment of platform trials versus conventional trials in terms of resources, time?
It's a great comment. It has not been done to my knowledge. We are building a cost effectiveness capacity into REMAP-CAP, but that's going to be looking at the individual interventions, not the model itself. It probably is cost effective, but there are additional costs with the platform, and I think it would be a very smart thing to do. The one other comment about the costs of this, I am increasingly of the view that these should not be funded by peer review funding agencies, that they actually should be funded out of operational funds from the healthcare system, in the same way that any large initiative puts aside a portion of its funding for research and development, that this is really part of what the healthcare system has to do. That can give it some longevity. It means you're not kind of going from grant to grant. And it also gives the healthcare system some input into the priorities and the questions that get asked in it.
Yeah.
John, that's particularly relevant for transfusion medicine, where the blood products are coming for free to the hospital. And so you really have like a central agency providing and paying for them. And now we're going to start to test them in areas where we have no idea whether or not certain transfusions are helpful. Yeah.
Well, it's really interesting. You could imagine a transfusion platform that the essence was simply a product going out from the Canadian Blood Services. Any product that was run by Canadian Blood Services would be eligible. So you might then have multiple different things, from immunoglobulins perhaps to factor concentrates, depending on the disease, and those could be the different domains for the trial.
So just following up on that idea, I mean, I think, everyone I think loves the idea of sort of operationalizing research. That's just part of patient care, is, and embedding it that way. I guess the thing I always sort of struggle with, in considering, these very nimble, adaptable trials that you can kind of get going right at the beginning of something you don't fully understand. You can move quickly and generate data very quickly. On the other hand, there is this other model of drug development, where you have your phase 1 trial, then your phase 2, and your phase 3, all of which is meant to ensure that you fully understand what your intervention is, so that you don't, there's so many examples of trials that probably went to like phase 3 clinical trials too soon, and failed, and as a result kind of died, and there was a missed opportunity there. And similarly, when you have a condition where you don't really even understand the disease well, if you jump in too quick, are you going to miss an opportunity? I was really thinking a lot about your reference to the Lynch studies of scurvy. The other thing about scurvy that kind of really held back discovery for that disease was that there was this huge debate about whether this was an infection or not.
Yeah.
And, you know, there was so much debate about this. I'm just sort of wondering, how would you, if you could go back in time, how would you apply some of these larger, platform study trial designs to address a question like scurvy when people couldn't even agree what the disease was, let alone, I mean, he had no idea what was in lemon juice. Someone had tried it once, and the idea about vitamin C, no one even knew what that was. If we have a disease nobody understands and a treatment nobody understands, can you sort of go into like a platform design and hope to, be able to understand what it is you're coming out with? Or do you need to kind of sit and do that basic science, work your way up, and, I guess you could argue that so many people died of scurvy because, we didn't just sort of go in and start studying, treating it right away. But what do you do in that situation where
Well, so I mean, there's a bunch of things in that question. The difference between different phases in clinical trial design. I mean, at the start, ideally, you would know a lot about the biology before you started trying to manipulate it in the context of a clinical trial. The reality is we don't. I mean, we use steroids all the time, and we don't really know what steroids do in a bunch of different diseases, but we do know that in some they change the course of the disease. So it's not, and with COVID-19, we knew very little about it at the time that we actually started the trial, so that it became a way of we could study the trial without having that kind of knowledge. So I don't think that doesn't preclude running a study. You can, though, you use the example of vitamin C thing. If that were a platform, with response adaptive randomization, what would happen would be you'd start off with a lot more sailors drinking barley water and hydrochloric acid, and very quickly you'd find that they were all taking citrus fruits because that was the one that was getting them better. And so it would happen without knowing what it was. They were just lucky that one of the interventions happened to treat the disease. That was a matter of luck. In the drug development arena, though, platform trials are being used in oncology in breast cancer. There's the I-SPY trial, which is being used to identify candidates in the early stages of drug development that should proceed to phase 3 clinical trials. So they have criteria for graduation and dropping them. And that model's been used in the GBM Agile trial as well. So there is a role in the early phases of drug development. To me, though, they're big. Where I find them most exciting is in this comparative effectiveness or phase 3, phase 4 type research, where you know pretty much the pharmacology of the intervention, and you're just trying to find out if it works better. And again, the platform trial can actually answer some of those questions. So you can use biomarker stratification, and that biomarker stratification will help you to understand the biology going forward. You can collect samples, and we're putting in capacity to do that into REMAP-CAP, so we can use it as a discovery platform as well.
Yeah, just follow up on that. Like, let's say, in the middle of REMAP-CAP, somebody had come up with some root extract and they said, like, this seems to work. And then you plugged it into the adaptive trial design and, and it looked like it worked. Would you be obliged to then follow it up with a traditional sort of phase 3 clinical trial? Or is the statistical robustness of a platform trial enough to say, look, this is now enough to apply for licensure? Like, what's, like, do you have to sort of go back and kind of do the traditional, drug development methodology to get licensure?
I mean, I think that's more of a regulatory question. FDA does accept platform trials as part of the drug development process. So I think that would be the case. Obviously, something that's quite out there, what their decisions about what additional studies might be would be open. But we actually had some experimental drugs on the REMAP platform. We had a TLR4 antagonist and a couple of compounds, blanking on the names right now, but it didn't work out all that well. There's additional challenges in oversight of the trial, and the relationships with the pharmaceutical companies who wanted to have access to the information were difficult to work out. So we didn't really get very far with that. Still kind of keen to look at that and potentially use REMAP-CAP for phase 2 pharma studies, but there are a whole host of other complexities. We're not even a blinded trial, for example. We don't blind our interventions because, you know, we've got too many of them and they're going to be variable. So that I think is a little bit down the road.
RT, you undid your camera. Do you have a question? No. Okay.
Can I ask, or I guess to ask a question and make a comment? These platform setups are very tied with Bayesian analyses in the sense that when you're doing adaptive randomization, there's a kind of an unusual paradox that the number of individuals in the treatment arm at the end of the day is actually informative about the treatment effect.
Yes, exactly.
So conventional analyses are, they're not going to give you unbiased estimates of treatment effect.
Yeah.
So I just wondered if you had any experience in sort of meta-analyses, or just thinking about if there was a sort of a requirement to do a more conventional phase 3 trial to support a finding from a platform study, if you ever had experience with that.
So it's a fascinating question, and the answer is no. I mean, we were doing it just because the need was there. So this is kind of under fire, but those are really important questions. The response adaptive randomization seemed like such a great idea, but it's complicated. And we've been burned on a couple of occasions where we've had, once when we moved our data center, and for a fairly long period of time, we were operating on an old randomization scheme, which was actually preferentially randomizing people if something was harmful. That was in the vitamin C domain. And we just recently had a situation where we had a coding error in the corticosteroid domain, which meant that the RAR, it didn't change the outcomes, but what it changed the response adaptive randomization. And so at one point we had it that up to 90% of patients could be randomized to one arm. And that was a mistake. Because as you say, sites were saying, how come, you know, everybody we're recruiting is getting this? And obviously that meant they were probably unblinded. So I think there are some really important issues there that need to be thought through, whether it's using a more conservative approach to response adaptive randomization, whether it is a corollary frequentist validation, when those things should be done. Again, when you're working in a comparative effectiveness thing, I think it's probably less important. If you're introducing a new therapy, it may be more important that you have that kind of conventional validation. But I think these are all fascinating questions that need to be answered in the future.
Great, thanks.
All right, I'm afraid we are out of time. Raz, I saw you had a question in the chat there. I would encourage you maybe fire that to Dr. Marshall, if that's okay, by an email, and because I think that's a good question about you know, how to, you know, convince a grant reviewer to understand, you know, a proposal for Bayesian analysis.
They're getting better. They're getting better at
Okay.
CIHR. They're usually pretty good about that. It's been my finding.
Good to hear that. Okay, well, listen, we're past the hour, so I will I'll let everybody go. Again, thank you so much. It's always so thought-provoking, this topic, and you know, good for the transfusion community to get more familiar with this.
Great.
Okay.
Okay, thanks so much.
All the best. Thanks.
Take care, everybody.
Thank you.
Thanks. Bye-bye.
Bye. Take care.